स्टार्टिंग से करूँ? हाँ स्टार्टिंग। एक मिनट। वो कोई थैंक यू करो। Myself, Raj Kumar Sharma, lecturer Bio at Government Senior Secondary School, Gonakara, District Hamirpur, Himachal Pradesh. Today, we will start a new chapter, that is Biotechnology, Principles and Processes. First of all, we will try to understand what is biotechnology. Biotechnology is the technique of using live organism or their enzyme for products and processes useful to humans. The European Federation of Biotechnology defines biotechnology as the science and organisms, cells, parts thereof, and molecular analogues for products and services. By using biotechnology, we prepare the different products which are very useful to human welfare. So, how these products are formed by using the biotechnology. Biotechnology is a newly emerging science in which we correlate the various subjects like physics, chemistry, and bio. Biotechnology deals with the micro-mediated processes like making curd, bread, wine, etc. It also deals with in vitro fertilization, commonly known as the test tube baby program. It deals with the synthesis of using of a gene. It also deals with the preparation of DNA vaccine and correcting a defective gene. So, biotechnology deals with various types of fields. Next, the principle of biotechnology. Some core techniques of modern biotechnology are used. These core techniques are the genetic engineering and bioprocess engineering. In genetic engineering, we use those techniques in which genetic material, DNA or RNA, is chemically altered and introduced into organism to change phenotype. While in bioprocess engineering, there is the maintenance of sterile ambience in chemical engineering processes for growing desired microbes, eukaryotic cells in large quantity for manufacturing of antibiotics, vaccines, enzymes, etc. In genetic engineering, we follow some basic concepts in genetically modifying an organism. These steps are the identification of DNA with desired gene, then introduction of identified DNA into the host, next the maintenance of introduced DNA in the host and transferred DNA to its progeny. So, we will discuss about the first step, that is the identification of DNA with desired genes. The traditional hybridization techniques lead to inclusion and multiplication of undesirable genes and desirable genes. In traditional hybridization, some unwanted genes may be inserted during hybridization technique. In genetic engineering, only desirable genes are introduced. So it is a very precise technique by which we isolate the desired genes and then we introduce the desired gene into the host cell. Next is the introduction of the identified DNA into the host. A vector DNA such as plasmid or viruses are used to deliver an alien piece of DNA into the host organism. Alien piece of DNA means the foreign DNA or the DNA with the desired character. Look at this picture, this diagram. This is a bacterial cell. This is a bacterial cell, and inside the bacterial cell there is a main chromosomal DNA. Okay. In spite of this bacterial DNA, there are small stretches of circular DNA. These small stretches of circular DNA are known as the plasmids. These plasmids are used as the vector in biotechnology. Next step is the maintenance of introduced DNA in the host and transfer of DNA to its progeny. A piece of alien DNA has number of sequences called origin of replication needed for starting replication. So, it cannot multiply itself in the progeny cells of the organisms. Hence, alien DNA is integrated into the recipient genome. It multiplies and inherits along with host DNA. Here the recipient genome is the plasmid. The process of joining and inserting a foreign piece of DNA into host organism to produce new genetic combination is called recombinant DNA technology. The first recombinant DNA, also known as the rDNA, was produced by Stanley Cohen and Herbert Boyer in 1972. They isolated an antibiotic resistance genes, a piece of DNA from the plasmid of Salmonella typhimurium. It was linked with a plasmid vector and transferred into E. coli. As a result, the gene was expressed and multiplied in Escherichia coli. Now, the next topic in this chapter is the tools of recombinant DNA technology. We know that the DNA is very small in size. It is very difficult to see the DNA. So, to remove the desirable piece of DNA, we use various tools in recombinant DNA technology. The first tool of the recombinant DNA technology is the restriction enzymes. Another tool that we use in recombinant DNA technology is the cloning vector. And next is the competent host. So, three main types of tools are used in recombinant DNA technology. These are the restriction enzymes, cloning vectors, and competent hosts. So, we will study these tools in details. First of all, the restriction enzymes. Restriction enzymes are also known as the molecular scissors. These enzymes cut the DNA at desired site. So, what are restriction enzymes? These are the enzymes which cut DNA at specific site into fragments. They belong to a class of enzymes called nucleases. In 1963, two enzymes responsible for restricting growth of bacteriophages in E. coli were isolated. One enzyme added methyl groups to DNA, the other cut the DNA at specific site. More than 900 restriction enzymes have been isolated from over 230 strains of bacteria. Various enzymes have been isolated from the different microbes. So, it is necessary to classify them and to name them. So, how do we name the restriction enzymes? We use some patterns to name the restriction enzyme. The name of the restriction enzymes is started from the capital letter. This capital letter is taken from the genus of the microbe. The next two letters indicates the species of prokaryotic cells from which they were isolated. For example, ECO R1. The ECO E letter, capital E letter, shows the genus of the prokaryotic cell, that is Escherichia, and next to small letter indicates its species, that is the coli. So CO letters are taken from the coli. And R stands for the restriction enzymes, and the Roman numbers indicates the sequence of the restriction enzyme in which it was discovered. Now we have studied that restriction enzymes belongs to a special class of enzymes called nucleases. These nucleases are of two types: exonucleases and endonucleases. In exonucleases, they remove nucleotides from the ends of the DNA. In case of endonucleases, they cut at specific positions within DNA. In the exonucleases, as we have studied, these enzymes cut the DNA from the ends of the DNA. These are the ends of the DNA. So the exonucleases cut the DNA at the terminal sites. But in case of endonucleases, these enzymes cut the DNA within the stretch of it. The first restriction endonucleases is the hint second. It was the first restriction enzyme which was discovered. It cuts DNA by recognizing specific sequences of 6 base pairs. This is called the recognition sequence for hint second. This is the DNA sequence, and the hint second enzyme cuts the DNA sequences at particular site. So these are this region is the specific site which is recognized by hint second. Restriction endonuclease recognizes a specific palindromic nucleotide sequences. It is the sequence of base pairs that read the same on two strands in 5' to 3' and in 3' to 5' direction. When you read the base pair, from 5' to 3', like GAATTC, from left to right direction. When you read another strand of the DNA, the same nucleotide base pairs are present, that are GAATTC. So these are the palindromic nucleotide sequences. I again repeat, it is the sequence of base pairs that read the same on two strands in 5' to 3' and in 3' to 5' directions. Restriction enzymes cut the strand a little away from the center of palindrome sites. What between the same two bases on opposite strands. This leaves a single standard overhanging stretch at the end. At the ends, they are called sticky ends. Suppose this is the DNA stretch. Here are the nucleotide base pairs. Okay. So restriction enzymes recognizes the DNA stretch, its base pairs, and cut the DNA in such a manner that two sticky ends are obtained. These are the sticky ends. Enzymes cut both DNA strands at the same side. From left to right, it recognizes the side GA from 5 prime to 3 prime, and also recognizes the same base pairs from 5 prime to 3 prime. Sticky ends form hydrogen bonds with their complementary cut counterpart. This stickiness facilitates action of DNA ligase. The DNA ligase is such a enzyme which is used to ligate the two strands of the DNA. Ligase means to join. So this enzyme is used for the ligation. When cut by the same restriction enzyme, the DNA fragments have the same kind of sticky ends. They are joined by DNA ligases. This is the foreign DNA. This foreign DNA is treated with the restriction enzymes. Okay. The same restriction enzymes digest the plasmid DNA. As we have studied that we are using the same type of restriction enzymes to cut the foreign DNA and the plasmid DNA. Our main motto is to join the foreign DNA with the plasmid. So the similar sticky ends should be formed. So to get the same sticky ends, we treat the foreign DNA and plasmid DNA with the same enzyme. The next tool is the cloning vector. It is a DNA molecule that can carry the foreign DNA segment and replicate inside the host cell. So, this is the bacterial cell. This is the main chromosomal DNA of the bacterial cell, and these circular rings are the extra chromosomal DNA called the plasmid. These plasmids are the circular DNA molecules which carry 10 to 30 genes. So, the various cloning vectors are used in the recombinant DNA technology. Some examples are here. The plasmid, bacteriophages, yeast, yeast artificial chromosomes, or BAC, bacterial artificial chromosomes. These plasmids or bacteriophages are used as a cloning vector. This is the virus which grows on the bacteria, so they are known as the bacteriophages. Bacteriophages are those viruses which grow on the bacterial cells. Bacteriophages has high number per cell. This means that they have a very high copy number of their genome within the bacterial cells. You know that the viruses have the protein code, and inside the protein code genetic material is present. When such virus infects the bacterial cell, then it introduces its DNA into the bacterial cell. Inside the bacterial cell, the virus DNA replicates and forms thousands copies of viral genome. This is the virus. This one is the bacterial cell. And replicated copies of viral DNA. When the cloning vectors are multiplied in the host, the linked piece of DNA is also multiplied to the numbers equal to the copy number of vectors. The next topic is the features required for cloning into a vector. In a cloning vector, there should be some features that each vector must have the origin of replication. The vector should have the selectable marker or the marker gene. It should have the cloning site. The cloning vector should be fit for plants and animal cells. This is the structure of a cloning vector, which is known as PVR322. In this cloning vector is present inside the E. coli cell. So each vector must have the origin of replication. It is a sequence where replication starts. A piece of DNA linked to origin can replicate within the host cell. It also controls the copy number of DNA. So for getting many copies of target DNA, it should be cloned in a vector whose origins support high copy number. The cloning vector must have the selectable marker or the marker gene. Marker gene is a gene that helps to select transformant and eliminate the non-transformants. If a piece of DNA is introduced in host bacterium, it is called transformation. Such bacterium is transformant. If transformation doesn't take place, it is non-transformant. Selectable markers of E. coli include genes encoding resistance to antibiotic like ampicillin, chloramphenicol, tetracycline, kanamycin, etc. Normal E. coli cells have no resistance against these antibiotics. The next feature is the cloning site. To link the alien DNA, the vector needs a single or very few recognition sites for restriction enzymes. More than one recognition site generates several fragments. It complicates the gene cloning. So look at the E. coli cloning vector PVR 322. This is the DNA. This is the DNA stretch. Okay, and different genes are present in this cloning vector, or simply is it the plasmid. Okay, so different genes are present on the cloning vector, like a gene for origin, a gene for ampicillin resistivity, and a DNA segment on which CLA1 can be acted. The next, that is the tetracycline, gene for tetracycline, which have the recognition site for MAMH1 and CEL1, and the gene for ROP, which have the recognition site for PBU second, and so many other sites are present for various restriction enzymes. Ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistance genes. For example, ligation of foreign DNA at WAMH1 site of tetracycline resistance gene in vector PVR322. As a result, the recombinant plasmid is formed. If ligation does not occur, it is called non-recombinant plasmid. When a foreign DNA is inserted within a gene of bacteria, that gene is inactivated. It is called insertional inactivation. Here the recombinant plasmids lose tetracycline resistance due to insertion of foreign DNA. When the plasmids are introduced into E. coli cells, three types of cells are obtained. The first one is the non-transformants. They have no plasmid, so they are not resistant to either tetracycline or ampicillin. Second one, the transformants with non-recombinant plasmid. They are resistant to both tetracycline and ampicillin. Transformants with recombinant plasmid. They are resistant only to the ampicillin. The recombinant plasmids can be selected out from non-recombinant ones by plating the transformant on ampicillin medium. Then the transformant are transferred to the tetracycline medium. The recombinants grow in ampicillin medium but not on tetracycline medium. What non-recombinants grow on medium containing both the antibiotics. Thus, one antibiotic resistance gene helps to select the transformants. The inactivated antibiotic resistance gene helps to select recombinants. But this type of selection of recombinant is a difficult procedure because it needs simultaneous plating on two plates having different antibiotics. So alternative selectable markers have developed based on their ability to produce color in presence of chromogenic substrate. In this, a recombinant DNA is inserted into the coding sequence of an enzyme that is beta-galactosidase. So the gene is inactivated. This inactivation is also called the insertional inactivation. Such colonies do not produce color. These are identified as recombinant colonies. If the plasmid in bacteria have an insert, it gives blue-colored colonies in the presence of chromogenic substrate. So this is the culture of the transformant and non-transformant bacterial cells. The blue color dots shows that such bacteria are non-transformant, while the white colored spot shows the transformed bacteria. Next feature is the vectors for cloning genes in plants and animals. Genetic tools of some pathogen is transformed into useful vectors for delivering genes to plants and animals. For example, the agrobacterium tumefaciens, it is a pathogen of many dicot plants. We may use the retroviruses. Retroviruses are actually those viruses which contain the RNA as the genetic material. The agrobacterium tumefaciens can deliver a piece of DNA, that is transformed DNA, to transform more normal plant cells into a tumor. These tumor cells produce the chemicals required by pathogens. Tumor-inducing plasmids of agrobacterium tumefaciens is modified into cloning vector, which is not a pathogenic but can use the mechanism to deliver genes of interest into plants. This is the plant cell. This is the plant cell in which we have to introduce the agrobacterium tumefaciens. This agrobacterium tumefaciens carries the transformed DNA. The transformed DNA is that DNA in which we have the ligated the desired piece of DNA. We use another vector, that is retroviruses. As I have already told you that retroviruses are those viruses in which RNA is present as a genetic material. Retrovirus in animals can transform normal cells into cancer cells. So they are used to deliver desirable genes into animal cells. Since DNA is a hydrophilic molecule, it cannot pass through cell membranes. So the bacterial cells must be made competent to take up alien DNA or plasmid. For this, the bacterial cells are treated with a specific concentration of a divalent cation. For example, calcium. So DNA enters the bacterium through pores in cell wall. The divalent cation, that is calcium, increases the permeability of cell wall. Such cells are incubated with recombinant DNA on ice. Then they are placed briefly at 42 degrees Celsius and put them back on ice. This enables the bacteria to take up the recombinant DNA. There are other methods to introduce the alien DNA into host cells. Nowadays, we use the micro-injection needle, gene gun, etc. The micro-injection recombinant DNA is directly injected into the nucleus of an animal cell. Ballistic gun or gene gun is also used to introduce the transformed recombinant DNA into the host cell. Cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA. This method is suitable for plants. Disarmed pathogens, vectors, are also used to introduce the recombinant DNA. So, we have discussed about the biotechnology, their principles, the different tools that we use to get the recombinant DNA or to get the desirable genes. In tools, we have studied about the restriction enzymes, the various vectors like plasmids, bacteriophages, and the host cells which we use to introduce the desired DNA. In the next session, we will study about the processes of recombinant DNA technology. Thank you.